Good evening, ladies, gentlemen, and non-binary friends. My name is Elijah St. Pierre, and I will be your presenter today. We're going to be exploring some long-term experiments going on the east coast of New Hampshire, specifically in the Great Bay and also at the mouth of the Piscataqua. This research has not actually started yet. It'll be starting next semester, and it'll be with myself, Dr. Elizabeth Harvey from the UNH Phyto Lab. So sit back, relax, and enjoy the show. We're going to talk about some of the focuses of this study. We are focusing on marine prokaryotes on the coast of New Hampshire, of which we're focusing on two different types, cyanobacteria and heterotrophic. These bacteria are diverse in size, shape, and function. They all have different kinds of metabolism, and they're so incredibly diverse that it's hard to identify them underneath a microscope. Or at least if you think about a vial of seawater, it would be pretty difficult to pull out every single prokaryote in that vial. Here are some examples of just general, more diversity here. Really just some photos to show you guys some kind of secluded organisms that aren't necessarily all together. These are separated. We can easily identify what they are. I did not write down what they are, so let's move on. But one thing I did write down is about the bacteria that causes illness with oysters. If you guys have ever had a raw oyster, there are definitely some warnings out there about being infected by Vibrio fulnificus. This thing could potentially kill you, and unfortunately, it shut down quite a few oyster industries over the years. This is found in marine estuary environments, and in Great Bay we do have at least one oyster farm going on there. So this is something we would want to monitor for. Another thing that we should be taking a look at is some of the bacterial blooms that can occur. This is just an example in the Gulf of Mexico. I was wiping out some fish and being incredibly toxic. This is back in 2018. So why does any of this really matter? Well, in coastal New Hampshire, we do have increasing levels of chlorophyll that are occurring, and that chlorophyll is from bacteria that is able to photosynthesize. And obviously, if that chlorophyll is increasing, then we want to know what kind of bacteria are going to be present and if they could be harmful to us or to the ecosystem. One thing we really need to be concerned of is that sometimes the composition of chlorophyll really matters more than just the chlorophyll level. You could have high chlorophyll levels, but until you understand exactly what that organism is and how they kind of chain together, this is an image of, I believe these are diatoms chaining together here. That gives us more insight into any kind of complications that may occur or just the type of organisms that are present, which is very important. This study is going to be focusing on some of the community changes that are occurring at two different locations here, the Coastal Marine Lab and the Jackson Estuary Lab. We're going to be using a bioinformatics pipeline in order to take a look at some of this data, generate the data, and hopefully, not hopefully, this is going to be one of the first longer term experiments of this quality for Great Bay. These are some images of Coastal Marine Lab and Jackson Estuary Lab. So the Coastal Marine Lab is right at the mouth of the Piscataqua. Piscataqua is this big river that comes through and divides Maine and New Hampshire. And the Jackson Estuary Lab is right in Great Bay. These are just some more photos of the area. I do not have a photo of the Jackson Estuary Lab, but I do have a photo of the Coastal Marine Lab, which is in the upper right-hand corner, and a photo of the Piscataqua. That's actually the Piscataqua whale. Back in 2024, a whale was just in the Piscataqua, and I happened to be on my boat with my dad and my uncle, and we had the privilege of seeing that whale, which is a very unique sight, being in the river and not just off the coastline. This is examples of some process data that we have over the previous year. This is not what is currently being processed, but stuff that has already gone through processing. And this data will be used in the future to kind of construct different graphs and conduct different tests. In order to process through the bioinformatics pipeline, we're going to be using Michigan State University Aviti Illumina short-read sequencing sample requirements. They have a whole pipeline, and that's what we're basically going to run through to follow their procedure for our bioinformatics part of this experiment. Some directions we're going to be looking at in this experiment is changes in alpha beta diversity, and really the important part here is you want to have a better understanding of the composition of bacteria in Great Bay. This may not go down to the species level, but it can definitely go down to the order level, and that's really what we want to try and understand. Currently, I am still in the process of extracting DNA from the hundreds of samples that we have from the Jackson Estuarine Lab and also the Coastal Marine Lab. I've processed about 186 so far, but there's at least another 200, if not 300 more samples to be extracted, and especially because these are in replicates of 3. They're basically pulled on a specific date, and we have to go through and conduct some qubit testing on them to ensure that there is some form of DNA concentration in there before we go through and process this through the bioinformatics pipeline. Once all this data has been processed through the bioinformatics pipeline, I'll be using RStudio to really take a look at some preliminary data and then developing a hypothesis off that, and then generating a very similar final presentation, basically discussing some of the interesting findings of that data. The goal is that this will be a long-term running experiment, so years and years and years from now, this should still be ongoing, and hopefully we'll have like a decade worth of data that we can really take a look at and potentially make some predictions off that, or at least monitor those communities for the future. Thank you all for taking the time to watch this presentation. I appreciate it. Please do not use this for any other means than for Population Genetics 705-01. This is not an official presentation of any means. This was thrown together relatively last minute and is not for any kind of official use. Thank you all for your time. I really appreciate it. Have a good day.